Following Griffith's famous experiment on Streptococcus pneumoniae, three other scientists, namely MacLeod, McCarty, and Avery, they continued the experiment on the bacteria and they fractionated the heat-killed S type into three different components, namely polysaccharides, the proteins, and DNA. And in their experiment, the first set of experiment, they removed the polysaccharide capsule of the heat-killed S type and injected it along with the live R type into the mice, and the mice died because the transformation was there, and the R type were transformed into virulent S type, and they developed pneumonia and they died. And the second set of experiment, they removed the protein fraction from the heat killed S type and injected it along with the live R cell type into the mice, and again the mice developed disease and they died. The transformation was not affected. And in the third set of experiment, they added the enzyme proteases into the heat killed S type and injected it along with the live R type into the mice, and they again developed pneumonia and they died. And the last set of experiment, they added the enzyme deoxyribonuclease. This is the enzyme which destroys the DNA, and they added it along with the live R type and injected it to the mice. And the mice, there was no transformation of the R type into the S type, and the mice remained healthy. So, proving once again, it was the DNA. Once the DNA was destroyed, there was no transformation of the non virulent R type into virulent S type, so no pneumonia, and so the mice remained healthy. So it was again, it proved that it was the DNA which was the transforming principle of Griffith. This DNA transformed the